talk. As Joachim mentioned, there are 3 major scientific goals that we are following in this network. And the first one is about viral genome sequencing and microbiome sequencing. And Stephan Ossowski will talk first. Let me just introduce him a little bit. So he is a professor for clinical genome analysis and a group leader for computational genomics at the Institute for Medical Genetics and Applied Genomics in Tübingen. His title is Estimating the Mutation Rate in a Host SARS-CoV2 Population by Ultra-Deep Sequencing with Unique Molecular Barcodes. So share your screen, please, and the floor is yours.
I hope you can see my screen now.
Yes.
Good. Thank you for the introduction. Good afternoon. And I'm glad I can present our work today here. Thank you for the invitation. I'm presenting today on behalf of the viral genome team of the Decoy Consortium. So we are all concerned with the sequencing of the virus and also the microbiome from swabs. And we have several major contributors apart from the NGS competence network centers that were already mentioned. We are also having the TU Munich, the Robert Koch Institute, and the Helmholtz Center and Max Planck on board. So there it's quite a strong collaboration. And I would say we have a very heterogeneous sequencing landscape, but our goal is to build capacity to sequence more, to share our protocols and to exchange material. I also want to say that we are very open to sharing our data with the community via, for instance, GIS8, and we are open to new members. So please join us. We meet almost weekly via video conference. And quickly, the funding sources, our main funding source was DFG, but there are some other contributors. I'm sorry, I have some hiccup here. I quickly start that again. So we have the contributors to the German viral genomes on the GIS8 website. More than half of all the viral sequences on GIS8 come from our consortium, as you can see here. And most importantly, the leader of our work group, Alexander Dilde from Düsseldorf, they have really contributed a vast majority of these sequences. And I'm sorry for that hiccup here. But a major problem that we still have is, unfortunately, that despite our efforts, Germany is on the very low side of contributions to GIS8. We have a very low fraction of the German samples are actually sequenced, and that is something we want to change. Why is that so important? If you go to the Next Strain website, which is using the GIS8 submissions, the viral genomes are used to identify novel clades. For instance, here, the clades that have been identified in Europe. So that can be used to identify novel strains that might actually be potential risk, for instance, for the vaccination, or could also be hard to detect by current RT-PCR approaches. And as you can see, these are here the divergence, the number of variants between the different strains. And we are also already reaching 37 here. That's better visualized in this way here, also from Next Strain, where they show over time, the last 9 months, that there's a linear increase in variants in the discovered strains, now reaching 37 here. And these are only the SNPs that are clonal in the patient, so meaning that they are shared by all the viruses of a specific patient. And this is not possible here to discover subclonal variants that are just found in a low fraction of the patients. And that is what we want to discuss today. So the decoy sequencing of SARS-CoV-2 genomes is quite heterogeneous because we have so many centers, we have many approaches. All of us have to do some steps like reverse transcription, obviously, from RNA to cDNA. But already here, the library preparation differs. We have amplicon protocols and we have oligohybridization-based protocols. There are different amplicon protocols, and we have many different sequencers from nanopore to Pacific Bioscience sequels to all the Illumina sequencers. So there's quite a diversity, and I want to discuss today specifically our approach in Tübingen using the oligo enrichment. In the beginning, we first tested if there is a difference in the yield using different reverse transcriptases. And we did actually see quite a difference based on the virus load. Here, example from 1 million to 1,000 viral copies per microliter. And we could see that some of the products give much better yield than others. So this way we could decide on which ones to use. And as I mentioned, in Tübingen, we decided to go in my institute for the oligo-based enrichment instead of amplicon. This was already introduced in the first session by the Twist representative. Here we use oligos that are complementary to the virus genome, to the cDNA, and then we can use magnetic beads to capture these viral sequences so that we strongly enrich them compared to all other like human or microbiome DNA that could be in there. Finally, we sequence. This product, the Twist SARS-CoV2 panel, can be found here. It was implemented in my institute by Michaela Pogoda, and these are our collaborators from Twist, which I want to thank here. Then we sequenced the first clinical samples. We also used synthetic viruses from also from Twist to test different copy counts. And we also made synthetic mixes where we could simulate SNPs. Again, I want to specifically thank our clinical collaborators from the ICU, Michael Bitzer and Siri Göpel, and also from the virology, Tina Ganzenmüller, Daniela Betztan, Angelika Ifter, and Professor Iftner, who have provided all these samples. One can say here that the yield, the sequencing yield and the coverage are basically mainly depending on the copy count of the virus. You can see that here with the synthetic mixes, and that is exactly also seen based on the cycle threshold of the RT-PCR. So the lower the cycle threshold, the more original virus material was there, and the higher is our yield. So it's important to choose samples with a low CT value. That also shows in our coverage profiles. So here are the synthetic mixes. With 1 million copies per microliter, we get really nice coverage here. And then the lower we go, the less coverage. You can see here only 10x left, and with only 100 copies, there is very little we can sequence. Same, here's a comparison for the amplicons, where you can actually see that with similar copies, you get more gaps than with the oligo enrichment-based approach. Here are the clinical samples. They look very similar. Lower CT value, much better coverage, which we can use to identify low frequency variants. And we obviously uploaded samples and virus sequences to GIS8. And as you can see, even in the small city of Tübingen, we already have 4 different virus strains represented. So there must have been multiple introductions of the virus to Tübingen. Now, for the second part to the discussion, I wanted to start about measuring the evolution, the intra-host evolution of the viruses. So the host has a specific virus population. With a low CT value, we would assume there are lots of viruses, and we can extract lots of viral copies. Once we have reverse transcribed them to cDNA, we can sequence them very deeply. And if we now assume that there is 1 or 2 or 3 viruses who have a specific mutation, so it's a tiny fraction of them, but it's there, we would like to actually detect that fragment and see that there's a mutation within all the other non-mutated virus sequences. And for that, we want to go to an extremely deep coverage, which obviously is only possible if we have low cycle threshold. So if you have lots of viral copies, otherwise that's not useful. But still, we know that Illumina sequencing has a high error rate, relatively high. Nanopore has an even higher error rate. How can we actually find something that's only 1 in 10,000 or 1 in 100,000? The trick is unique molecular barcodes. And here we work together with IDT and combined the adapters, the UMI adapters from IDT, from the XGen protocol, which was also introduced in the last session, with the Twist SARS-CoV2 panel. And the idea of unique molecular barcodes is that each original DNA fragment is barcoded, and after PCR amplification, you can now exactly know which of the sequences are copies of the same molecule. And that can be used to correct errors, like here where there's a sequencing error. But if there's a true change here from A to C, you see that all of the sequences show this A. Well, we tested that on our synthetic mix here in a 1:99 mixture, and it worked very well. In that mix, we expected these 4 variants. One of them is an indel, and we can see here that we could detect these 1% T, 1.5%, and 2% easily. We then went on to sequence, as I said, extremely deep. The deepest we got was 1.1 million. And then we have to deduplicate, so we have to merge these PCR duplicates of the same molecule. That will lead to the true coverage, so to the true unique molecules, which you then see here, which is about 30,000x. And during that process, we can correct the errors. As I said, here we can correct this G here and this G here. And for that, we place these in groups. So the more copies, the better the correction. So for instance, for 2 copy barcodes in that experiment, we had 7,000x coverage where things were represented twice. And we had about 8,000x coverage for molecules that were represented 4 times in our mix. And obviously these have higher quality. They have better, stronger error correction than these ones. You can also see that these errors are specific for the nucleotide change, but with 4 barcodes, with 4 copies, we can correct them all to basically 1 in 40,000 error. And with that at hand, we can now make statistical models that measure how many minimum number of alternative reads do we need to actually be sure there is a variant. And that again differs based on the numbers of copies we had. But with 4 copies, you can see that there are already no errors left, no positions in the genome that have 3 errors at the same position. All of that we put into a pipeline called UVVAR, you can see here, and we even added other variant callers, not just our own, but VARScan, NoFrag, and IVAR, and added our error correction. And the result is this. Here you see the allele frequency of detected SNPs and the p-value. You can see then here all the clonal variants. They are 100% in the population. Here you see samples that are a mixed population, so they are 50-50 variants. Then you have all these low frequency variants that must be novel in the patient. And if you zoom in here, there's quite a lot between 1% and 10% going on here. Very interesting. Depending on the depth we reached, that was the one with 1 million depth, we could find up to 200 variants per patient, but most of them are below 1% frequency. If we take all our 32 patients together and check the distribution of variants over the whole genome, we see that basically all positions of the genome can be mutated. These are 100 base pair bins. There are a few hotspots, but not really. I mean, they are fairly random. Most of the changes occur except for G to C. That's super rare. That's the mutational signature. And finally, we compared the 4 variant callers. And that is a bit shocking, but we already expected that from our experience in cancer. They do not overlap extremely well. And that's what we will have to work on next. As you can see, there are some variants they all find, but then there are also variants that are specific. For our own UVVAR, the majority of variants here, they are below 1% because the other 3 tools only call above 1%. With that, similar to Joachim, I want to thank really a lot of people here, mainly the people that worked on the viral genome sequencing. Special thanks to Alex for leading our work group, Michaela for implementing this UMI protocol, and the virology and ICU from Tübingen who collected the samples, and my bioinformatics people for implementing UVVAR. Thank you very much.
We thank you, Stephan, for this really interesting presentation. And I already noticed a question from the audience. This is from Maria Bukhari, and she is asking about the diversity of the identified samples in Germany and if they are all equally lethal. Can you comment on that?
Yeah, that seems to be a question for a clinician. I don't know if they are all equally lethal. It is quite diverse. I mean, as I showed already in Tübingen, there is quite a diversity. And in Europe, we have the most diverged samples I could find on next strain is from Europe. It's from London. So I think in Europe, due to the density of our cities and the travel, we do have a very, very diverged population. But if that already has an influence on the, yeah, I don't know, on the survival rates or things like infection rates, I don't really know.
Okay, thanks. There's one other question I can ask from Matthew Peasant. Are you incorporating UMIs during cDNA synthesis? If not, would you see an advantage in doing so?
The details of the protocol you would have to discuss with Michaela. We create cDNA first, and then we add UMIs. Our UMI protocol we originally developed for liquid biopsies for cancer monitoring, where we obviously start with DNA. And so here it's very similar. Obviously, there is this one step we cannot correct errors for, which is the reverse transcription. And anything that happens until we can add the UMI. So there would be definitely an advantage. We would, yeah, we should get a slightly lower error rate because we can correct this one extra source of error. I think you are muted, Peter.
Yeah, thank you once again. So in the interest of time, we have to go on, and our next speaker is